the phospholipid membrane, we have phosphate group, and then you have literally two lipid molecules hanging off it, and the phosphate group is very hydrophilic, right? And the lipid groups are very hydrophobic, right? And they line up, and you get an outside the cell, and you get the inside the cell, and this really creates a nice insulating barrier between the inside and outside of the cell. And there's some evidence that there's what's called aquapores, which actually allow water to go through, but generally you have to actually open up some kind of channel between the inside and the outside to let anything go through. And there's also a lot of pumps which are actually actively keeping the inside and the outside to be very different in their concentration of ions, which ends up creating a potential difference. And so we will go through the equations talking about all of those. So here's the axon. The axon comes off and then can branch out, and it sends out lots of connections to generally, a lot of times it will go a long ways and then branch out. Sometimes it will just branch out locally. But it's pretty much a very highly, once an action potential starts here, the density of ion channels, voltage-gated ion channels, almost guarantees that action potential will travel down. And the density of ion channels are really highly balanced to maintain the minimum amount of energy while maintaining the maximum probability of the action potential reaching the end, too. So it's an interesting problem to keep that. So the dendrites are really where you get spectacular differences between cells. And this is where you can really see some cells look very different from others. So on the left, these are paintings or drawings done by Ramon y Cajal, who is a Spaniard that did lots and lots of anatomy, some of the original anatomy. And he won the Nobel Prize, one of the early Nobel Prizes in neuroscience for this anatomical work. And here are different drawings he has of, so here are the different cortical layers. So this is layer 1, layer 2, 3, 4, 5, and 6. And you can see here's an example of a cell body here in layer 5, whereas this one has a cell body in layer 3. Both these guys would be called pyramidal cells. And different layers where their cell bodies are and their dendrites look different too. So there's some information passing in layer 1, so you see this, what's called a tuft, or a bunch of dendrites that sort of sprig off at the top. And then here are those basal dendrites down there. And these are called spiny neurons because when you really zoom in, you can see these little bumps coming off of the dendrite. And the spines are an indication of actually, that's a postsynaptic specialization of the dendrite to meet up with the presynaptic. Not every cell is spiny. It turns out mostly excitatory synapses are spiny, whereas inhibitory synapses tend to be non-spiny. And one of the nice things about spines is that it dramatically increases the amount of surface area. If you have to receive 15,000, sometimes up to 50,000 synaptic inputs onto you, that takes up a lot of surface. And one way to do that is to make little bumps so that you increase the surface area. The other is that these spines look like little, they look like little mushrooms. And so they go down to a very small head. And so whenever these neurotransmitters open up all these ion channels, well, you have a lot of interesting feedback to the plasticity of the synapses are activity dependent. So if that synapse is active a lot and receives a backpropagating action potential at the right time as it receives a synaptic input, then you actually get a certain amount of calcium, which then affects the ion channel of the neurotransmitter receptors. And you don't want that information from this axon coming in and its firing time to change the plasticity of the firing time from another neuron next to it, for another presynaptic neuron. So what you do is you essentially encase that synapse and keep it literally chemically separated from everybody else. So you go through just a small little neck for the voltage to pass through, but chemically they're isolated. These are just examples of pyramidal neurons. And so the pyramidal neurons are sort of, there's only a couple excitatory neurons that you see in the nervous system. The pyramidal neurons are sort of your workhorse. And somebody said that, the pyramidal neurons, it's like the granite. I'm trying to see if I can get this right. It's like the granite, but the inhibitory cells is like chiseling away. The inhibitory cells are the ones that shape everything. And therefore you have all of most of the variability you see in cell types are amongst the inhibitory cells. And so, we focused on pyramidal cells mostly, and the inhibitory cells are sort of junk. And now we recognize that really most of the diversity in the nervous system is coming from the inhibitory cells. So, well, I mean, even Cajal could have told you that. But, the importance of it. Here he's actually naming glutamate. Glu is the amino acid that it's releasing. So that amino acid is the excitatory neurotransmitter. It's the primary excitatory neurotransmitter in the brain. GABA is the primary inhibitory neurotransmitter in the brain. Glycine is the primary inhibitory in the spinal cord. And so here are different GABA, and they're non-spiny, so you can see they're different in their morphology, and they release different neurotransmitters. There's just a tremendous difference. Like, I think this one is in the retina. Some of these, like this one here is one called a chandelier cell. It is hard to see here, but at the end of each axon, it actually makes a whole like 10 or 15 synapses onto a single cell. So you will see one of these chandelier cells literally synapse right here and send 15 synapses, inhibitory synapses right here. And so when that neuron, so you could have all this excitatory input coming from different areas, right? And then it comes down and, summates and then causes the cell to fire. But if the chandelier cell wants to turn that cell off, it opens up all the channels here and this cell does nothing. It shuts that cell down. And they have really just a neat morphology. The surprising thing is, and this is sort of, I shouldn't say things like this yet. Even though that one happens to be inhibitory, it turns out that the chloride reversal is flipped right at that area because the pumps are actually different there, and those actually become excitatory, even though it's a GABAergic cell. So these are where things start to get really weird very quickly. You have your sort of canonical story of inhibitory, excitatory, and then you find out in development, even the inhibitories are excitatory at some developmental ages, and even in adult animals, sometimes inhibitory cells are excitatory, and even in conditions where, like during epilepsy, we think the inhibitory cells sometimes turn excitatory. So nothing is ever written in stone. So here is the synapse, sort of a blow up of the synapse. Here are the vesicles. So these are the little bubbles of neurotransmitters sitting inside the cell. And then this cell, when an action potential comes down here, triggers it through a calcium signal to release the vesicles and the neurotransmitters coming out. And you see these little patches of receptors, and often these receptors will be co-localized with the release sites. So it's not just a random patterning down there. But and then the ion channels open as the neurotransmitter hits it, and then ions flow in, which change the voltage of this cell, and then that voltage sort of propagates down the dendrite. So here are some reconstructions of a dendrite. So, with nice high-quality confocal microscopy, not only can you see a single cell, but you can create a 3D reconstruction of it. One of the questions we have is, when animals start learning, you'll actually see increased number of synapses. And often you'll see double synapses between one cell, presynaptic cell, and a postsynaptic cell if that becomes a really important connection. And one of the questions is, well, where does that double, is it the one synapse splitting into two, or is it them forming a second connection? And I've seen some images where you can actually see the two connections between those two cells and another axon or dendrite passing between the two, which indicates it's not just one that split into two, because then there would be nothing between it, but one and then it formed another connection and there just happened to be something in between it. So this level of anatomical details is what we're getting into and is really telling us a lot. We take brain sections and slice it up in really, really thin slices, and each slice looks like this. You can see each one of these are nuclei from the different cells. These are probably mitochondria, the much darker things. This, to me, looks like a big mumble jumble of membranes and intracellular space, and very little extracellular space. But then you can take a whole series of these things and using some very sophisticated image processing techniques to register all of these images, actually then reconstruct different axons by following from slice to slice to slice, reconstruct different axons here. So axons are not nice, thin, cylindrical things. You can see they're actually get thickened in places where there's connections, and then thin, and then get thick again. And then here you can take the 5 or 6 of them and reconstruct them in space.